What's up, humans, and welcome to a new Psicoactivo. Today's episode, I have Miss Alaina Hardie and Mr. Alan Izarraras. They are experts that are doing right now work on the Nazca Mummies DNA. But I want to give them the microphone to introduce themselves and tell me what the expertise are and what is going to be presented today. Let's start with Alaina. Welcome, Alaina.
Thanks. It's good to be here. So I'm a data scientist. Probably like close to 13 years ago, I went to Singularity University. And I was there like computing stuff. And somebody introduced me to molecular biology. And then someone else was like, oh, you can do computers and molecular biology together. It's called bioinformatics. So that's how I got interested in bioinformatics. So I kind of, taught myself to do that. And I co-authored a book on genetic testing with my company in 2017. Yeah, and so that's how I'm here. I mean, like my day job is as a data scientist, and I work with a lot of AI and ML and do a lot of, genomics stuff as part of that. So
Okay. What
uh,
about you,
yeah.
Alan? Welcome to the show, Alan.
Thanks, Paul. Glad to be here. I am in academics. I'm in academia. I am a PhD student last year. My field of interest is population genetics, or more fully known as computational population genetics. So I work in a lab that studies human genomics, the human genome, and yeah, I have some expertise in ancient DNA and overall interest in human evolution.
Okay. So, Alaina, how did you become interested in the Nazca mummies? When did this first drop on your plate, so to speak?
I was browsing Reddit and I came across a post that was talking about this, and I saw someone mention DNA, and I sort of clicked through some links and I saw that there were like sequencing runs uploaded to the sequencing read archive for the NCBI, the US National Institutes of Health. And I was like, oh, and people had made like certain claims about what this stuff said and didn't said. And I was like, oh, well, I can actually show people you can do this yourself. Like that this isn't magic. It's just like typing commands. So that was my idea, is that I was curious in the question, like, can this tell us anything about non-human life? The data are there. So I thought, well, I'll do this. And I found a collaborator on Reddit, and we wrote a report after doing, I mean, what is effectively like really basic, like a metagenomics and human genomics pipelines, and wrote a report, and that's how I kind of stumbled into it.
What about you, Alan?
Um, I had heard about these Nazca beings, I think from documentaries in History Channel, but it really caught my attention when Maussan brought them to Mexican Congress. That was when my mind started racing and I just couldn't stop thinking about them because the evidence was a lot more robust. And so starting doing research, discovered there's a sequence actually uploaded and say, hey, this is exactly what I do. I have this expertise. I can do things. And then, yeah, one thing led to another. And that's how I contacted Alaina, who I think is the only person that had sort of um, written documents of what she had done.
Okay. So it's safe to say, I don't know, where do you stand before we start with the presentation? And I know this is kind of a loaded question, but I would like for you to maybe give me a as honest an answer as you can give me. Where do you stand with these bodies, specifically the M types, because those are the ones that you guys have investigated, correct? So, where do you stand with those? Do you believe these were once beings that lived and had tridactyl features in them, or do you believe something different? Let's start with Alaina.
So I think that they were once living beings, the M types, absolutely. In fact, I think the J types were also once living beings. The story is more complicated with them. I am agnostic on the question of whether the tridactyl feature is is like part of development, or it's post-mortem, or even pre-mortem. Like I don't think there's any, I haven't seen any convincing evidence that would let me say any of those things. And I think that the space in which these DNA samples, the sequencing runs are non-human is a very shrinking space the further you look into it. I think that's how I would characterize what I would say right now.
Okay. Alan?
Well, I like to stay fairly open-minded. I don't want to conclude anything. I haven't made up my mind. All the time I switch what to believe, but they do look incredibly compelling. I mean, the moment I saw the 3D scans of these beings, it's something that's near impossible to replicate, like to fabricate, I mean. And so I guess the question remains if, okay, you could go with the extraterrestrial narrative, right? Or it could also be the case that it's just a different population or a new species of hominid that hasn't yet been described. So I remain open to the possibilities. I think ultimately these are definitely interesting. It's definitely real. I mean, it's tangible. I haven't seen the bodies, but the 3D scans are remarkable. And for that reason, they should be studied and seriously by experts, by people with expertise and multiple expertises, not just the DNA side, but all sorts of fields.
Yeah, as you guys know, I've been talking to anthropologists, biologists, forensic scientists. So I do agree with you. I also think that the case didn't start off on the right foot, if you ask me, because there's been a lot of stuff happening in the background and around it that has stained the case in many ways. And that's made it a little more difficult to try and honestly and openly investigate this. So hopefully that we can get after this presentation, we can get a little bit more clarity, and so you can share what you have studied and the important points that you feel that people need to know about. So can you please start sharing the presentation so we can start, and whomever wants to lead it, just let me know and I'll just shut up and let you guys do your thing.
How about I share the screen and you talk, Alan?
Sure. You can also
Okay.
chime in whenever. Okay, so yeah, let's just introduce the samples that we do have. So in NCBI, which is this global repository for genomics or genetic data, there are 3 samples uploaded. Two of them are purported to come from the same being, which is Victoria, which is one of the small beings. And we also have Maria, which is, I guess, the hybrid, some people call it, and it's called Ancient 003. So we have supposed carbon-14 dating. These are 790 years ago and 1,700 years ago. So at this age, we definitely expect them or these sequences to have ancient damage. So that's something important to keep in mind. We also could do mitochondrial haplotype, so we can look at all the mutations of the mitochondria. The mitochondria is this really small DNA molecule inherited by the lineage of mothers, and we can recover sort of these haplogroups, which are related to a certain evolutionary pathway. And so what the program shows is that we recover haplogroups. Well, first things first. When dealing with these samples, one also has to wonder, are these done in good faith? Are these really coming from the samples that they're reportedly coming from? And
Oh,
also,
you mean like provenance? Yeah, you mean yeah.
exactly, yeah, because we have to act in good faith, right? We don't know if this is like some sort of elaborate hoax. We don't know if they are deliberately contaminated. So in ancient DNA, contamination is super common. When dealing with anything ancient DNA, one has to go to a specialized lab. One has to wear these full suits. One has to clean the room. These labs have, for example, negative air pressure, so the air only goes out and it cannot go in. And since when dealing with human samples, this is especially relevant because we ourselves, the people who manipulate the samples, have DNA everywhere, right? So any mishandling, it's very, very easy to get contamination. And so here is uh, a data point that contamination might be the case because we have the same sample, which is ancient O2 and ancient O4. They are supposedly come from the same being, but they report different mitochondrial haplotypes. Also relevant to mention, the mitochondrial DNA is the most common DNA to have contamination from, because mitochondria are in multiple copies in every cell of the body. So we have a mitochondrial haplogroup of H27, which is related to Eurasian populations, and of M20, which is related to Southeastern Asia. So that already tells me that there's probably a degree of contamination. We don't know to what extent. It's very hard to tell, but this is the most likely explanation. And then we also have Maria, which also is reported to have the M20 mitochondrial haplotype, also associated to Southeastern Asia. Anything you want to add, Alina?
No, I think that captures it.
So apart from these 3 samples uploaded to NCBI, Alaina did some researching and we also found other runs. Do you want to talk about them?
Sure. So these were done at IPN, I believe. And so this was the first set where they didn't get a lot of good DNA. Well, it turns out those runs have been uploaded and are just sitting on a Dropbox, and they're linked from, I think it's from the Abraxas report. So just for comparison, we did go and grab those. And there are some interesting things that we discovered through a friend and collaborator of Alan's, that I think are kind of interesting features of this. But the thing to keep in mind is that they're very low quality sequences, right? There's not a lot of reads in them, and they don't look ancient, which is, I think, the thing that's most interesting. So, Alan, do you want to take over and talk about like why we think that might be from that first group?
Yeah. Well, first of all, they have the DNA contained is very small, right? We only have so in DNA sequences we have 2 categories, right? We have total number of reads that were sequenced, but then of those total numbers, if you want to compare them with, say, the human genome, which is the case, you do a mapping. So that's the category for mapped reads. So only 7,000 of those 341,000 in the case for Mumia 1 were comparable to human sequences. It's a way to put it, right? Which seems like a lot, but it actually isn't. And so going back to what I was saying about the ancient DNA and that we have statistical ways of telling if this DNA looks ancient or not. We have the next slide. So this is called map damage. So the main characteristic of ancient DNA is that at the very start of the reads, we have mutations that typically go from C to an A. And so at the start, exactly, yeah. At the start of our graph, we are looking at how many of these missing corporations we encounter. So and this is what we got, right? So the upper graph is the ones we got from the mummy samples, which is these are really weird looking. In my experience looking at these graphs, it's very uncommon that we have a bunch of missing corporations in the middle. What we should be seeing is what's on the on slide number 4.
Something more coherent, and this is the one in the top is not as coherent. It's just like all over the place. But what does that mean in layman's terms? That
yeah, let me just show in slide 4, which is actually the example. So this is what it should look like. So at the beginning, a high number of these missing corporations, and in the middle of the sequence, flat. That's sort of the hallmarks of ancient DNA. So that just means that at the beginning of the read, we have these C to T mutations, which are associated with ancient DNA. So this sort of confirms that we are dealing with ancient DNA. And if you go back to slide 3, we don't really have that signal. There's no high missing corporation at the beginning, and there's like very, very noisy.
What does this tell you? That it's not ancient, but what else?
Yeah, it's essentially that this does not look ancient.
Is this Maria? Is this Maria?
I believe the one in the bottom is Maria.
Okay.
One of those lines is Maria. But neither Maria nor Victoria do they exhibit what we might call ancient DNA.
That's weird.
So, yeah, exactly. That's weird. And there may be different reasons why, right? It could be that the carbon-14 dating was done in maybe in the Diatomea sand. Maybe it wasn't actually the being, right? Maybe it was an associated thing. That's a case. Another case that I'm like sort of, if you try to be generous and open-minded, it could be the case that these beings were so well preserved that DNA didn't actually go through that process.
Doesn't diatomaceous earth preserve things like for a long time? That's like its function, right?
Yeah, but it's unknown to what extent, or at least it's unknown for me. There are plenty of ancient DNA published studies from all sorts of sites. For example, the most popular or the most high yield DNA come from what's known as permafrost. So this is like really, really cold temperatures. And even then, DNA is fragmented.
Okay.
So for me, it doesn't make that much sense that this mummification process or whatever they did to these bodies would yield better results than, let's say, permafrost. But it is a possibility that maybe the way these beings were mummified and dried or whatever process they happen to have could have saved the DNA from having these common associations.
Do you think that their biology, and this is a question for Alaina, I think, more likely. Do you think that their biology could be a factor that because they're so different from us, if they're not human, that when they were desiccated, they went through a different process of desiccation and it preserved them? Could that be a factor, or or
It's
not?
interesting. What I actually think is an interesting version of that question is why something that didn't develop in our on our planet, in our biosphere, why it would have the same marker, like mechanism of evolution that we do. So I think it's worth asking if you're going with the alien hypothesis, like why we're even talking about DNA and why we're even trying to answer those questions. Like I tried to like illustrate this or really early on by saying like I have a doll. My daughter has like a little stuffy, right? Okay, and I rub my hands all over it, and you sample that and sequence the DNA, and you're going to get like an Anglo human, right?
Yeah,
But her stuffy is not an Anglo human. It doesn't have DNA. Like it's like made of polyester.
yeah.
So it's the wrong tool for the job. So I don't know what the explanation of this is. We did find something kind of like an interesting hypothesis with the IPN, the original four, which is that there's something there are a whole bunch of like chemical processes you do, right, to prep the DNA. And one of the things that you can do if you have DNA that's been kind of like banged around in the environment is that you can run repair on it. And it's just like a chemical process, right? If you run this process on the ends where damage tends to happen, it's possible that those ends are going to get chopped off or corrected, and you don't see the ancient sample. And it could be this is legitimate DNA from the organism, right? There are lots of different explanations for this, but I do think we should be asking ourselves the question, if we're taking the hypothesis of extraterrestrial origin seriously, which I think is a hypothesis worth considering, why they would have DNA in the first place. So
I can maybe, because this is something that I've thought about for a long time. That I think it's one of the hypotheses that I think it's might be plausible is that life is kind of the same everywhere in the universe.
oh yeah, I mean
And
that's a widespread like perspective in astrobiology, but we don't
yeah,
know.
yeah, we
Right?
don't know,
That
but
one of the reasons that this question is most interesting to me is that it is a question we cannot get an answer to any other way. I mean, I guess we could synthesize life from completely different substrates in the lab and answer this question. But until we find life somewhere else, everything that we talk about is just speculation.
yeah, because we
Um,
only have our own stuff to compare.
right. And I guess I just want to problematize any assumption that we might make that it is not even universal, but like more than just our planet or even our solar system. I think we need to, if we're really um, trying to take a step back and look at this objectively, I think that has to be something worth considering.
Yeah,
So even all of this DNA work that we've done, and I think it's really interesting, and we've learned some really cool stuff. It could be the wrong question. We could be asking the wrong question.
I think this is the big chunk of the whole presentation that we should consider. And the big news for me that um yeah, it's modern DNA. It's not really ancient. That's
Well, we
really,
don't know that it's modern. We can say that it does not have these couple of
yeah,
hallmarks
it
of
doesn't have
ancient DNA,
ancient
right? That's
DNA
different.
from here. Yeah,
Those are
yeah,
two
you're
different
right.
things. We're not saying it's modern.
You're right. You're
Yeah.
right.
I mean, correct me if I'm wrong, Alan. I don't want to speak for you.
It remains unknown. So
Yes.
yeah, that's the
Exactly.
answer.
Yeah, yeah. Also, people have to know that in science, um, we have, if we want to conclude something, we have to have evidence from multiple different experiments. So if you run independent experiments and they all show or suggest something, then we could take the leap and say, okay, this suggests that it is not, it looks modern, right? Um, Also, since in the field of genomics, it is since we are dealing with essentially data and very, very big sequences of data. Genome, I mean, the human genome is 3 to the 9 base pairs. Yeah, all our methods are purely statistic, right? So if you run a method and you change even a single parameter, artifacts are very common. So you can have like a really shiny results that's really striking, and you can go with it and then figure out it was simply an artifact of not running the statistical program correctly. So this is just to say that all this information must be really replicated and really looked very strictly. Like one should always try to doubt of first results and try to replicate it and corroborate it. And so if we also another data point to suggest that this looks like modern DNA, could you go to slide 2, please? Is the fact, well, when we look at Maria, ancient 003, we have so many reads. This is like the number of genetic data captured from the sequencing run is massive, and that you wouldn't expect from ancient DNA. Typically, from human ancient DNA, you are looking at the coverage, so that means that the sequencing run managed to cover all base pairs of the human genome. You're looking for like 0.1 to 1. I mean, 1 is like excellent, like it's extremely rare. What's more common is like 0.1, 0.2, even less. And here we have a coverage of 15 times. So that means that we have 15 reads for every position in the whole genome. So that's also where it's another point of evidence to say that this doesn't look ancient. This looks modern.
Okay. What about let's continue with the presentation, and then I'll ask you more questions, but this one was necessary because that really threw me off a little bit. Thank you for explaining.
Sure. Well, yeah, so far we have just looked at basic genome statistics, and right now, or the main result that we have is a PCA plot. Could you show it, Alaina? This was work done by Alaina, and so here, PCA is a dimensionality reducing statistic. So it associates what closely resembles a given genome, right? So we have 3 case genomes, which is the Victoria, two Victoria samples and 1 Maria samples. So we want to, this method separates them into what other genomes it looks like. So the dots that are colored are all from a reference panel of different human populations across the globe. And anything you want to add, Alina? Maybe you should talk about this.
Yeah, I mean, so basically what you do here is you have a giant reference set, and I use the 1,000 genomes reference set, which is like 2,500 different, like deeply sequenced individuals. And they're labeled with their superpopulations. So African, Americas, East Asian, South Asian, European, Pacific Islander. I don't think Pacific Islander is in this data set, but that's like a superpopulation, right? And then there are subpopulations. So inside of the population of the Americas, there is, for example, a population of people of Mexican descent from Los Angeles, people from Puerto Rico, people from Medellin in Colombia, people from Lima in Peru, in the Americas. So this plot is colored by superpopulation. So like you can see like how incredibly widely spread this African superpopulation is, right? And so this sort of brown color here is the population labeled Americas, and that includes the Colombian and the Peruvian, as well as the Mexican in LA and the Puerto Rican. So, and I can show you, like, in fact, if I switch my screen share, I can show you like a fancy 3D visualization where we can control all the colors and stuff. But effectively, what you see here is ancient 003. So this is Maria, the sample from Maria. And this is
Okay.
ancient 002 and ancient 004. And so if you remember from this previous, like these are both supposed to be from Victoria, but they're slightly different results. And I believe they were two samples from the same site. I'd have to go back and check, of Victoria. And Victoria was the seated headless J type,
Yeah,
right?
yeah.
One of the very first ones. So, and remember again that I'll just point out a couple of tidbits, and I don't know if it matters, but like they're separated in time. If these carbon dates are accurate, right, they're separated in time by like 1,000 years. And if you know anything about the human migration, you know that populations like come in waves and disappear. Like they're like things change, right? You know, there are empires, like there's lots of reasons that you might see different results. But the fact that these two, I would say, cluster together is interesting, especially because you expect them to come from the same remains. But both of these definitely plot in where you would expect people from that time period to plot in.
Okay.
And I do have, I have a comparison. I did the same analysis that we did on these three. I downloaded some results from a study from Chachapoya in northern Peru. And I did the same thing. And that's how, for example, we have comparisons of ancient DNA, like what ancient DNA actually looks like. Because I've done these exact runs for these other samples, which I know were from a published study on ancient DNA. And we actually, I mean, it was like this whole process was like super fascinating what we've learned. But I think this is the thing, like independently of everything else, what this shows is that the DNA that's not mitochondrial or Y chromosome, so like chromosomes 1 through 22, if you take all of these like million or more millions of data points, right, and you kind of like collapse them across this data set into these little points, their point on this chart shows you kind of like their relationship to each other. And so what you're looking at here is the relationship of Victoria and Maria to human populations in this data set. Did I get that right, Alan? Did I miss anything or misstate anything?
You nailed it. Yeah,
Okay.
that's basically it.
And
Also worth
I
mentioning.
understood it well too, so thank you.
Awesome. Also worth mentioning is this is a three-dimensional statistic, and this is only a slice of component 1 and component 3. Maybe we could look at the whole thing, but this captures the essential message, which is that these three samples, both Maria and the small being, fall under human genetic variation.
Okay.
And specifically, Maria seems to fall exactly where one would expect it to be, right? Which is in Native American ancestry, close to Mexico and Peru.
Yeah. So she's our relative.
She could be, yeah.
I want to ask something to Alaina that she told me at the beginning. Because there's been a lot of controversy about the smaller beings. And you just said something that surprised me a little bit, because you said that you believe that the smaller beings were also once living beings, but it's a complicated matter. And I see a lot of people that are pro Nazca Mummies going at it online. I've been even called a disinformation agent for trying to get a more objective outlook on both types of bodies. I still don't know if the smaller bodies were once living beings. I have been talking to Pioti, who believes they are because he says he saw one in the 90s. But I want to ask you, what makes you say that? I'm very interested to hear what you think about that.
Well, I mean, I think so. So first of all, you're asking the wrong person. Okay. So I'm qualified to have the conversation about like the numbers that we did here, right? So what you're asking me to do is to step outside of my expertise and share with you my perspective. So just qualifying that this is my
Yes.
perspective. I mean, I think it's very clear that these bodies are the J types are organic, which means that they were necessarily formerly living, right? I think that the sample that you took from Victoria was taken from the top of the spine, right? So you would expect that whatever the top of the spine was, that would be the same spine all the way down, right? And if you look at the imaging, this is what I've been told. I took these images to like the CT images of Josefina and Alberto, I believe were the two, to my friend, an anthropologist, without any information about what this is. And she looked at the images, like immediately looked at the images, the CT. I was like standing there with my computer. We were sitting outside the University of Toronto. And she was like, that's from a hominid that walks upright. And that's kind of intended to be a joke because there is only one of those right now. Like it's humans, right? Like we're the only hominids that walk upright right now. But the point is that like she could take one look at this anatomical structure and say, oh, like this is the expertise of an anthropologist. And so if I put that together with the idea that like you can take a sample from the top of the spine, which is where the spine from Victoria, the sample was taken from Victoria, and all of that looks like a cohesive piece in the imaging, you have to at least take the like ritual construction hypothesis seriously, right? That this could be part of a human or hominid who walks upright, who was placed in this position and then assembled.
Yeah.
And so I think one of the things that's really important to me is that like something that this showed us is that we should really take the concept that these are remains, like these, like the J types are remains, right? And the M types are remains, and they're remains of individuals who used to live. And I don't think, I think the claiming that the J types are like made out of cake or 3D printed or whatever, like that's complete, like it's totally falsified by the imaging. And so I think you have to take that ritual construction hypothesis seriously. And I think you have to take seriously the idea that they include human remains.
Okay. What about you, Alan?
What my opinion is on the small
Yeah.
bodies?
What do you think about those? Yeah.
Um, I think there's a possibility that they were legitimate beings that once were alive. Yeah, but it's unsure, right? I haven't seen the bodies personally. The people that have seen them seem fairly convinced. I mean, I don't know what else to say. It's certainly extremely interesting. I certainly hope it gets more attention. I mean, right now what we have done, we have definitely centered more towards Maria, the M type.
Yeah.
But that's simply because we're only two people, right? Like maybe, and we both have day jobs. So if we had more time, I would certainly love to explore uh, what are things we can extract from the small being. And yeah, I don't know. I think there's a good chance that they may have been living beings.
I want to ask Alaina, because you also said about the tridactyl features, and I think that's interesting to explore because I don't know if you remember, but back in November 9th, there was a hearing in Peru where two doctors that were at this hospital when they took the bodies, they presented a study where they said, if I'm wrong or you think I'm wrong, please correct me, but this is what I got from that Maria was and her tridactyl features were not manipulated, did not seem to be manipulated. And that's what I got from that because they did studies on her and Wawita. And Wawita was a mutilated human. That was in the study. But Maria, they said that the tridactyl features were morphologically coherent. That's the terminology I believe they used. Did you catch that, Alaina?
So I have heard that, and I have heard the opposite, like from the Benoit, who's that?
Who? Who?
Benoit, I think, was the guy who did the tendon analysis video. So I've heard, which I think has, like, there's been a lot of discussion about the methodological issues with that recently.
Oh yeah, Benoit.
Yeah, Benoit, that's
Yeah.
it. Yeah, so I think I've heard like both perspectives. I can drop something a little surprising for you, which is that I went to falsify this natural tridactyl hypothesis about 6 months ago, like saying, okay, well, let's test this. Like if there is our tridactyl features, then and it's developmental, then there should be something in one of the developmental pathways that would indicate that you would have 3 fingers and not 5. So I dug into the literature and I found a list of 50 genes that were associated with digit development in tetrapods. And that's like all creatures that have like 4 limbs, right? And I got this list of 50 genes and I went to Maria and I pulled out that list of 50 genes. And then I went and I annotated that, like basically with each of the mutations in it, I went and referenced a big database that is like, okay, is this mutation pathogenic? Is it known to be pathogenic in the literature? And so what you would expect, especially in a developmental pathway like that, is you wouldn't expect any mutations, right? At least you wouldn't expect any mutations that you didn't kind of see. Like it would be a little surprising, I guess I would say. And I found one. I found a pathogenic mutation in a gene called GLI3, and I can go into the details about this. And I was like, that can't be right. So I did it again, and I used a different method to call the variants, and a different method to do the annotation. And I didn't have that result. And I was like, oh, okay, well, this is just that whatever statistical weirdness that Alan was talking about. So I did it again with a third methodology, and I got the same result where there is a mutation in, I think it was 6 of the 16 reads that cover this like one spot on this gene. And so I was like, well, that's interesting. I wonder what this mutation is. And I went and looked it up in the literature. And there is literature of there being, there's a Chinese family that has a similar, like a same kind of mutation, different position, but the same kind of mutation in this gene. And the people in the family who had this mutation had a chance of having polysyndactyly, which is like more than 5 fingers, or like fused fingers. Like it's like a different condition than tridactyly, but and the people who didn't have the feature that in the family didn't have the mutation. And even more interesting to me is that this mutation you generally, if you have mutations that come from both the father and the mother, you're more likely to see like some sort of change. But this particular mutation only comes from one of the parents in this person. At least that's statistically what it looks like. And in the family, you don't need it from both parents. You only need one parent to have this missense mutation to pass it on to the child, and the child can show these features. So I'm not saying that we have discovered genetic evidence. I got to be really clear. We haven't discovered genetic evidence of tridactyly. We haven't discovered it, like I don't have any evidence that that's what this is. But I can tell you that that's like a kind of an interesting finding, and something you would expect to have 5 fingers and doesn't, right? Like you have to at least be like, huh, I don't know what this means when you put it together with all of our other evidence where it doesn't really look ancient, and there's like weird coverage, but like this does not, I did not find that in Victoria. In the Victoria ones, I only found it in this GLI3 gene in ancient 003.
Okay, so a couple of things about all of this, because one of the things is that it becomes clear to me that speaking to scientists who are trying to investigate this sometimes becomes challenging, because there are often people that have a bias. And I didn't find this with you two. I want to hear your thoughts on that. What do you think? Where do you think this bias can originate from? Do you think, um, because I've seen a lot of this phrase jumping the gun, specifically with this case. There seems to be a part of the scientists who are studying this right there in South America who seem determined to that the bodies are non-human, like they're saying they're non-human, and some of them even believe that they're from another planet. I don't think that is still the case. I think it's too soon still. And I know it's been 7 years. I mean, 8 now. I know people may be desperate there, and some of them maybe have spent a lot of money on this, their life savings, even. I don't know. Uh, their time certainly has been spent in this specifically. So let's start with Alaina. Why do you think that is, this jumping the gun situation that keeps happening?
I don't know. I mean, like, all I can say is it's not the way I am. Like, I can't really speak to the mindset of other people. I mean, I could create stories where I could explain this, but I don't know if they're accurate. I just, for me, I'm very comfortable in not knowing. Like, I'm extremely comfortable in ambiguity, and like the fact that I don't know means I get to learn, and that's really exciting to me. So I don't feel any need to like to have a position. Like, the process is interesting to me. The question is interesting to me. I don't
Okay.
know. I don't have anything else to say, I guess.
No, that's a great answer. What about you, Alan?
Yeah. The same thing that Alaina said. I mean, in these such contentious topics, you always have people that are more biased to a certain conclusion, right? Whether if it's aliens, if it's an ancient hominid, or if it's a fabrication. Yeah, what Alaina said, people are dogmatic. A lot of people really want to believe a certain narrative. I would encourage everyone to just place yourself in the mystery. Allow yourself to be open to new conclusions. Don't try to. I mean, it's like having your mindset of it is very comfortable, right? But just try to sit in that unknown. Like, reality is deeply mysterious. There are many things going on that we don't know anything about. Don't try to run with a conclusion just because a smart person said so, or you heard a smart argument online. Like, think for yourself, browse the evidence, um, listen to the evidence, but also uh, evidence. There is no super evidence, right? There's just like chunks of interesting bits of data, such as a trade, as the gene associated with different number of fingers that Alaina mentioned. And there are things that are interesting that pop to mind. Try following those, but certainly stay open-minded. And also, I wanted to say that it's been a blessing meeting Alaina, and the fact that she also happens to be one of these open-minded individuals that isn't that doesn't necessarily like want to it be aliens or to it be ancient hominids or whatever. She's open to the mystery, and I think that that's what makes for a great researcher, and
Yeah,
for a great scientist.
I agree. Both of you are same way. So that's really refreshing in this day and age. Before we move on to more speculative stuff about UFOs and all this stuff, I want to ask you, what type of collaboration would you expect moving forward from other people who are likely interested on this? And do you plan to publish any of this anywhere? Um, let's start with Alaina and then Alan.
Um, well, I mean, I think we would both love other people who are interested. Yeah, and yes, we would love to publish. I don't. This is kind of Alan's idea, so I don't really want to steal his thunder. But like he has said from the very beginning that we should be putting this on bioarchive, which is like a preprint server. It's like the most. That'll be the most effective way for us to get feedback from other qualified researchers before we go to publish it. I would personally love to publish. Alan's published before. I never have. I think that would be a cool experience. And what we would, I would love a couple of things. I will personally ask if you want to do this research and you want to use the stuff that I have done. All of my stuff is on my GitHub. You can download my pipeline. You can produce these PCA visualizations yourself. You can reproduce all of our work because it's done with using like really standard bioinformatics tools. If there's anybody who wants to contribute and has questions that they want to ask, that's great. The other thing I would ask is if there's anybody out there who has data. Like I know that there's like this side and that side and this, but like all I really care about is asking the questions. And so I am happy to ask the questions of any data that comes up. And if you don't want me to ask the question, just go to my GitHub. You can ask the questions using my tools of your data.
Yeah, and also another thing that I want to add to that is that something that happened, you're not going to bring it up, but I will. And this person already apologized for what happened, but your data was used in a paper that without your permission, and it was used to embellish stuff about this study. And I think that cannot happen, especially with such complicated cases as this one. So I would ask for the scientific community to please leave their bias at home. And if you're going to use someone else's or try to use someone else's work, you have to be in communication with that person and ask permission. And if you don't get it, don't use it. It's just standard practice. Sorry, but I had to say that. Alan, what about this publication? This idea that you're having, so let us know.
Yeah, for sure. So first of all, just understand that in academic science, these topics are completely like cuckoo, right? Like you aren't being taken seriously at all if you touch anything of this. So my objective is to do as thorough an investigation as possible. I've experienced publishing papers. I am becoming an academic. So I want to do this as thorough and rigorously as possible, so that any maybe more open-minded scientists or people in my field or related fields can look at this and like, oh, what this crazy Nazca alien mummy stuff, and see the paper and see, oh, well, they're actually doing things right. They're taking the correct steps. They know what they're doing. They know what they're talking about. So, okay, this is interesting. And I think by publishing this, hopefully this goes to the hands of open-minded scientists, academics, and this thing grows, right? Like this thing receives attention, and we can have answers and conclusions sooner than later that way. Because right now it's only me and Alaina. But hopefully maybe this podcast or this publication or whatever we do in the future can get in the hands of someone interested, and we can get in touch, and we can surely, both me and Alaina, I think, are happy to grow the team to welcome more people in. If there's anyone listening that has some knowledge in DNA, genomics, bioinformatics, or even data science, and is interested in this, we welcome that, and it will be awesome to build a team and have more people look at this.
Yeah, I will get you in contact with them if they contact me, for sure.
Mm-hmm.
That was a nice segue to talk about something that I think I want to take advantage of, because you both are scientists and we seem to be approaching a singularity moment. I did a video yesterday about that this dude, entrepreneur Mark Anderson, recently did an interview where he said that the government basically accepted that they have been hiding science and classifying science since like the 50s. And I want to ask your perspective. What would you feel or what would you do in this situation if you found out that governments and private corporations have hidden breakthrough science from the mainstream scientific community? What would your response be? Let's start with Alan this time.
Well, I mean, governments lie all the time. I don't think that that's news to anyone. And they certainly have an incentive to lie about or to prevent information from coming out about sensitive topics, such as potentially, military grade technologies or military use technologies. And so certainly what I'm doing is speculating, but again, there's many data points. There's many interesting things one can look at historically and even now in the present with all the UAPs and all the sightings and people that have come through with testimonies. I mean, there's a lot there. We don't really have like specific or a real conclusion, but the evidence is there and there's certainly something going on. And I don't think it's too wild to consider that that's the case.
What about you, Alana? Would you grab a pitchfork and a torch and march?
Pavel, I have a pitchfork in the closet back there. Like, no, this is I am really big on, I think I come from the open source world, right? I am really big on open information and collaboration. And I think that science is successful only when you get a bunch of people like banging on the same thing from different perspectives. And I think like hiding what I mean could conceivably be like developments in fundamental physics.
Yeah.
Is like, I mean, yeah, I would have a pitchfork. Absolutely.
Yeah.
Would you like one?
Of course.
I have a spare. Yeah.
Yeah,
I think it's outrageous. And I mean, I agree with like it's very clear that like governments lie to protect their own interests. They lie to protect national interests. Humans are basically, like I don't know. I think that it's entirely plausible, and I think we should all be outraged. If there is legitimate science that can keep us from hitting 3 degrees of warming and it's being hidden from us, that's not just like, that's a crime against humanity. It's a crime against the planet, right?
Yeah.
And so I don't know if this is true. I'm open to the idea it is. Like there's definitely enough smoke there that makes me think, yeah. And I would just I guess I would just say that if like there's a lot of stuff, there's something going on, right? And if they're hiding technology from us, from the human race that could like keep us under 3 degrees C of warming, or or all of the other things that, I don't know, gravitic propulsion could unlock for us, like that's a crime against humanity and it's a crime against the planet, and we should all be outraged.
Yeah.
If this is actually true, I might need that pitchfork. Like I really might.
To add on that topic about being outraged or not, I think I'm beyond outraged, honestly. It's just like a normal thought to me. Like it's just very pervasive, and I don't know what to think about it. I'm no longer. I no longer have emotions. I already have too many emotions. So
I want to ask you guys one specific thing before we go, because I can see that some mainstream like science communicators, right? Like Carl Sagan or Bill Nye, the science guy, or Neil deGrasse Tyson. You can tell that those guys are like actively trying to discredit any of these topics that seem more fringe and don't go according to the status quo of science, mainstream science. Those I understand. And if this is confirmed to be 100% true, that Science is being hidden, and we have been interacting with something over millennia that we still cannot explain what it is, but it's there. There is also a certain part of the scientific population that don't believe this is true and are like married to the idea that we're alone in the universe. Do you think that I happen to think that the ontological shock in them will be much greater because they've been living a lie for a long time. And how do you think that would go with for example, fellow scientists that you collaborate with that may be married to this idea that aliens are not real and we're alone in the universe and all that stuff? Let's start with Alaina first.
Well, I think scientists are people. That's one thing I will say. And so we come, like everybody comes with a set of biases, you know. And I have definitely seen in my time around science, I would say, I have definitely seen people who are very, very stuck in their ways. And I think that I don't know, there's a degree of intellectual arrogance that I find off-putting in some of these like big personalities like Neil deGrasse Tyson, that it's just like, I don't know, Gary Nolan always says, you know, you can only say this stuff as long as you aren't aware of the body of evidence. And so it's really hard to take seriously the opinions of someone who's like not at least familiar with the body of evidence. And I don't know, I guess that's what I would say. Everybody's going to have their own opinions, you know.
What about you, Alan?
Yeah, I mean, we're all people. We all have our identities. These guys, they have built a persona. They have built an identity. It's hard to give that up overnight. And even for the regular academic or the regular scientist, these topics they are allergic to these topics. They avoid them at all costs. And also, I wanted to add that there's this thinking around disclosure that is sort of going to be like a big boom moment and everything's going to explode and the world is going to notice overnight. I don't think that's going to happen. I think what is happening is that we are giving slow sort of drip information. There's people that have caught onto it. There's going to be a moment where the information is undeniable, where the data is undeniable, but still there will be people that choose to ignore it. And that's okay. And I think that is going to happen in many directions, not only in UAP disclosure, but also in technology, in spirituality, in psychedelics, in meditation. There is only so much that we can be aware of. And I think in all directions of human specialization, things are going to pop. And only a few are going to be aware of what really is going on. And the world is going to become wild.
Thank you for mentioning psychedelics. I agree with
you. I mean, these are topics that go hand in hand. There are many mysteries to this world.
Yeah. Alaina, and just before we leave, because I'm going to put you on the spot a little bit. I'm sorry, but I am. You went to Sol.
I did.
So there's a recent tweet from the Sol X handle where they say that they're going to release some of the videos. People are going to get early access, whatever that means. I want to ask you, out of all the conferences that you heard and attended, can you give us a couple of pieces of information that you thought were like, holy shit?
I don't think I really had any holy shit moments. I don't think there were any moments where I was like completely surprised or blown away. Like if I had been in sitting in the audience last year when Carl Nell did his thing, I would have been blown away. But Carl Nell just gave like a great, I mean, it was what was crazy. What was crazy was to sit up there, to sit in the audience, and hear these people from like Ryan Graves and you know, all of these people like stand up there and just talk about who's McKinnon, who's the guy from the Senate Armed Services committee. I think you,
Oh yeah,
the Kurt, Kurt,
McConnell, Kurt
Kurt
McConnell.
McConnell. Yeah. Like, here's a guy for 30 years on the Senate Armed Services Committee who's like, yeah, I'm pretty sure we have crash recovery. You know, like that was, I wasn't surprised by anything, but I was surprised by sitting there and hearing how pedestrian the conversation was around this, because it's like, these are like earth-shattering revelations, you
Yeah.
know? Yeah.
Okay.
So I guess I wish I could tell you like, oh, keep an eye out for like minute 13 of this presentation, but I can't. Like I just, what was the other thing I thought was really excellent was being around a bunch of people, smart people who take the topic seriously and are respectful of each
Yeah.
other. You know, it's like, if you want to tell people you're interested in this topic, you sound like a crazy person. Like, even if you're taking like the purely scientific perspective of like, huh, it seems like there's something interesting here, we should ask some more questions. Like, that's kind of unacceptable. So to spend a weekend in a group of people where that's not just like acceptable, but like it's encouraged to ask these questions and talk about these topics was really, really nice. And I found some cool collaborators. Like I've got this other project that I had kind of mentioned to you, where there are a group of us who are talking about building a system to monitor all of the air traffic control. You have these live air traffic control feeds, right? So the idea is we capture those feeds, transcribe them, we monitor them for mentions of like weird lights in the sky or whatever, and then we start logging that stuff. And if we put this system, Lester Nair had come up with this idea of like this open source system that's like this data repository, right? And we could like feed this stuff in, and suddenly you get like Seattle Tower talking about orbs flying out over Oregon.
Yeah.
And you can like activate a bunch of people to like monitor more air traffic control. Let's monitor Portland. Let's monitor Eugene. You know, let's do. And so I thought that was kind of a cool idea, and that sort of came out of Sol. So that seems really neat to me.
Nice. I like that. Lester is brilliant though. He's awesome. Okay, so I guess that's it for today. I want to thank you both for your time and for giving us this presentation. I'm gonna put it out later today, so I'll send it to you guys. But if you guys have anything you want to share in terms of where people can find you or anything you got going on right now, please share it. Let's start with Alaina.
Yeah, I mean, so I'll just link you to my GitHub project page where we have all of this stuff. You can see some visualizations. I'm actually happy to show you what one of the 3D ones looks like before we leave. And you can email me at verbalcant@gmail.com if you're interested in collaborating. We'd love to talk to you.
Okay, Alan.
Well, I have this other project. I like to think a lot about evolution at the grand scale of things, evolution of complexity. I do a bit of theorizing. I do that in my personal blog. I have some let's see, alternative models for evolution. And I also talk about that a bunch in my Twitter. So
I'll leave everything in the description when you guys leave. Give it to me. But Alaina, if you want to show me that before we leave, that'd be really
Oh, sure.
cool,
Sure.
so people can see it.
Can you see this?
Yeah.
Okay. So this is, remember I mentioned earlier that I got these ancient samples from Chachapoya in northern Peru.
Yes.
So what I did was I took one of those samples, and there were 8 or 9 of them, but I took just one of them, and I plotted it, did it exactly the same thing that I did in that chart, the plot that Alan showed you earlier. And
Yeah.
so that is this large dot. But remember how Alan was saying that there are 3 dimensions to this
Yeah.
data? Well, there's more than that. We're rendering 3. But you can see here that this is the population structure.
Yeah.
Okay. And I am coloring right now by individual population. And the original plot that Alan showed looked like this with the superpopulation colors. But like you can see, I can go in and I can turn off African, South Asian, East Asian, and European. And then you can see where this sample plots, right? Based on, so closest to this Puerto Rican population, it looks like.
So
that's
people
just
can
like,
access this on your GitHub?
yeah. You can just go
That's
to the,
awesome. That's
yeah.
awesome.
But let me switch to the other data set, which is the Nazca data set. And so this is what Alan was showing us before. And so there's ancient 2 and 4, and there's ancient 003. And again, so this is a light blue. Let's turn off some populations here. Okay, so Victoria, so this light blue is like close to the Colombian, Mexican, just like right there in the middle, right? But I thought that this was really useful rather than just like showing plots, because I think once you can kind of move around in here and see like how these things relate to each other,
Yeah.
I think this is a powerful tool for kind of like understanding what this represents.
Okay. Awesome. All right. Thank you for sharing that. And thank you again for your time. And if you like the content, please subscribe, hit the like button. I'm going to leave everything in the description, but I will see you guys on the next one. Thank you, Alaina. Thank you, Alan. Have a great rest of your week.
Thanks, Pavel.
Likewise, thank you.